All right, everyone. So now I want to focus on the walls of the digestive tract. Hopefully you've already watched the videos that talked about the pathway that you're traveling through and the fact that anything inside of that lumen is actually not in your body. Weird to think about the food in your stomach is actually not in you. But you have to cross that epithelium to actually be part of the body. So we should probably talk about what that epithelium looks like, how it's all organized, and the supporting layers that go underneath it, because it is actually very uniform as you move your way throughout the digestive tract. Now, I do want to specifically focus on my drawing for the small intestine, because that's where most of your absorption occurs. It makes sense to really talk about it, because there's some really cool features to the wall that help with that absorption. Basically, the idea being if you want to absorb a lot, you have to increase surface area so you can fit in more channels. And so you can slow things down. It's like adding speed bumps. And so you'll have folds that have folds on them. So we're going to draw that all out. We're going to start all the way on the outside and work our way in. And so all the way on the outside, if we're at the small intestines, is going to be the serosa. Now I did say we're specifically focusing on the small intestine here. So be aware of the fact that the term serosa is for any of the stuff, specifically the outer lining of the GI tract, in the abdominal area. So if it's around the esophagus, we don't use serosa. If it's the esophagus, that's going to be the adventitia. We end up with a different name because we have a different structural setup. If you're looking at the esophagus, it's not inside of a cavity. It's actually sandwiched between your lungs and your heart. So it's between the pleural cavities and the pericardial cavity in this place called the mediastinum. So as a quick refresher on what that all looks like, if you were to do a cross section, so let's say here's a lung, you're looking down on it, here's your heart up here, the mediastinum is this space here outside of those cavities. With the intestines and the stomach, the stuff in the abdominal area has a membrane that wraps around it. So it does actually have a serous membrane that it's going to be fusing with, and that's the peritoneal cavity. So we have the peritoneal cavity here, so we get a different name. So serosa for the ones touching the peritoneum, and then the adventitia is specifically going to be for the one up in the esophagus. All right, so serosa is our outermost connective tissue with some epithelium on it. Underneath that, we're going to start to have muscle. And again, we're going to have layers because there are some pretty cool features with this muscle. You can do peristalsis, so picture like you're kind of pushing behind it and moving it forward. You can do segmentation where it's going to pinch in different places along the ring, so you can break it into smaller chunks. And so you need to have muscle set up moving this way, but also muscle set up going like this. And so you're going to end up with longitudinal, so if we're making this as a big ring coming at you, we're going to draw the longitudinals as dots or circles. So we have longitudinal, and I'm going to spread these out, but realistically they're very, very close together. So we have a longitudinal layer of muscle. And if we're thinking about it, can we control this muscle? Do we have voluntary say over it? Nope. You can't tell food when to move from one organ to the next. You can't say, okay, I want to empty out my stomach now because that cake looks really good for dessert. It'll happen when it wants to happen. So it is smooth muscle. Then we'll have an inner layer that'll break things down into different chunks for us. And so that's where we can get our segmentation. So we have our longitudinal on the outside, and then circular, and I used a squeaky marker, on the inside. And so this is all made out of muscle, so we'll call this the muscularis. So the muscularis with their two layers here. So muscularis with an outer longitudinal, and then an inner circular layer. Now there is one place along the GI tract that gets an extra layer. Think about where you would need to have extra movement. In the stomach, because they're going to do mixing waves. It's a bigger organ, you got a lot of stuff going in there, so it's actually called mixing waves. We're going to kind of shuffle it all up in there. And so there's an oblique layer deeper here, where it would be at an angle. Now, your next layer is where we're going to first start to see some folding. And so our next layer is going to be below the mucosa. And so this, if it's below the mucosa, is the submucosa. This is the part where I was kind of telling you, start kind of think about like adding speed bumps. So this is going to slow food down as it passes through the small intestine. And so this is going to be the submucosa. And you can kind of picture like all this would be filled in, this connective tissue between that and the muscle. And so we have it here, kind of filling it in quickly. And so that's our submucosa with our first layer of folding. So submucosa because it's underneath the mucosa. So no surprise here, the next layer is going to be the mucosa. And again, it gets folds. And so the mucosa is going to have folds on it. We're going to call it the mucosa because this is the layer that will have the mucus cells. This is the layer that will produce that mucus and secrete it into that small intestine. It will also have simple columnar epithelium. So here it's kind of, and again, remember we're kind of coloring all this in. And also along the intestines, we do put some white blood cells and clusters here. Those are called Peyer's patches. That's part of your immune functioning. And so that is also going to be a feature of that mucosa. So hopefully kind of picture this and not have it look too awful for you. Should probably have taken an art class at some point. All right, so submucosa, then we have mucosa. And then on the surface of that mucosa, if you can kind of picture it, is going to be your simple columnar epithelium. So that's what makes up the top part of that layer. So this is with simple columnar. Remember, we're simple because you want to have the ability to absorb, but we're also columnar in shape because we want to be tall to make sure we're not absorbing pathogens, and also allows some processing as we bring things in. For example, like fats, we put a little protein coat around them. Now, simple columnar epithelium in the digestive tract can get some extra help for increasing surface area by putting on their surface microvilli. So they will have microvilli as their last little bit of folding. And that's where you can fit in the channels and the brush border enzymes. So folds on top of folds on top of folds. So you have microvilli, those are the folds of the cells. The submucosa, microvilli folds of the cells. The mucosa has their own folds. Then you have the submucosa, which are also folded. So hopefully that kind of helps show you some examples of how things would look through that GI tract. Now don't forget there are some differences depending on if you're wanting to do absorption. You'll use the simple columnar epithelium, but there are some places where you're not doing absorption, where you're gonna have some rubbing, like the mouth and the esophagus. So again, that's another place where the esophagus would be different. Instead of simple columnar, you would use stratified squamous, because if you get some rubbing there, you have other cells underneath to protect and also serve as backup if those top layers get shed off. All right, everyone, thanks for watching.